Now we're going to get into the eradication using chemicals. There are a lot of different things that we deal with. People think chemicals are nice and easy. They are not. They think they work great all the time. It only works as good as the person who is applying it, making sure that you're applying it properly. We have what we call disinfectants. These are things that you would use on things like your shovel or your pruning shears that could carry pathogen on them that you want to get rid of. We have what we call herbicides. Herbicides are materials that kill off plant material. With herbicides, we get rid of other plants that might harbor the same pathogen. We would do it alongside of the field. We might do it in the field. We're looking at ways that we can get rid of plants that might harbor that specific pathogen. We have insecticides. We use insecticides to kill off insects. Insects are vectors of pathogens. Sometimes the best way to stop a pathogen dead in its tracks is to make sure that we got the proper insecticide that we're out there killing the vector with. We've got nematicides. Nematicides are things that kill off nematodes. I'll tell you right up, these are among the most toxic of all chemicals that we find. Not saying that all nematicides tend to be very toxic. We do have some other ones that can be just as toxic as well. We have seed treatments. We always talk about treating seeds to make sure you reduce primary inoculum. Seed treatment is one of the cheapest and most effective methods that you can use to decrease pathogen. Therefore, it really should be looked at for virtually everything. We have soil fumigants. By fumigating the soil, we're going to kill off everything that's there. These work very well. One of the things they do is, even though they get rid of pathogen, they get rid of everything else. You might have a suppressive type soil that is going to suppress pathogen, but if you use a fumigant, it may not work anymore. We always have to look at pros and cons of all of them. Chemicals. First thing we got to deal with is toxicology. This is kind of an interesting story that we've got to understand. When they make these chemicals, they have to test the chemical to see how toxic it is. They have to see how toxic it is to the plant. They have to see how toxic it is to the applicator. If you've got something that's mega toxic to everything, you can't use it. If you spray it on and it kills off the plant, hey, it's no good. If it's so toxic that the applicator can't go anywhere near it, it's no good either. What they do is they study them in a manner by which they look at acute dosage, which would be a one-time dosage. How much dosage does it take, especially on the applicators? In this case, we're looking at the applicator. How much dosage does it take in one shot to severely injure or kill someone? They also look at chronic, which means you get a little bit of a dosage over a long period of time. And what does that do? And remember, we had those words carcinogen, mutagen, teratogen. We're looking at how toxic it is. In toxicology, they do these studies. Normally, they start out with these studies on something like, in the past, they used to use lab rats. Nowadays, they've got other biological modeling systems that they can do so that they don't have to kill off animals. Based on this, you can come out with different types of toxicities. In many cases, they need the mammalian systems in order to be able to see other things as well. They look at things that are oral, that are ingested. They look at things that are dermal, where they're absorbed through the skin. They will put on acute and chronic dosages orally and dermally to find out where they are. So this is really pretty scary the way they do it, and the answer is yes, it is. If you want something that is going to be relatively non-harmful to the applicator, we've got to go through these studies. If we don't, people are going to start dropping dead all over the place, and that is not a good scenario. They do these in these great big labs, in these great big buildings. They repeat experiments. All of that goes down to toxicology. In the end, what they are looking for is what we call the LD50. That is the amount of toxin necessary to kill off 50% of a population of test organisms. This is not what we want to kill as far as what we're using the material on. What does it do to organisms that might be there that are not what we want to kill? Now, this graph shows dose on the bottom, increasing off to the right-hand side, percent lethality going up. We've only got two ticks in here. We've got the 0 axis, we've got a 50%, and we've got 100%. when they start doing this, they gather this data, they find out what the percent lethality is at each dose. By doing this, they can figure out what is the 50% dose that kills off half of the population. We don't want to go on the extreme low end because that's something that could have died for any reason. We don't want to go on the extreme high end because these things take in much greater doses than we might want to look at. We look in the middle. That shows you where the LD50 is. What you do is you drop down and you find out what the dosage at that point. That gives you your LD50. The LD50 is looked at in a concentration of milligrams of material per kilogram of body weight. If we have something that shifts the curve over a little bit, that gives you a higher dose, which means it is less toxic. When they look at pharmaceutical products, they do the same sort of thing. They're looking at an effective dose. That's the amount it takes to be able to control a certain material inside of your body. They look at the lethal dose, which is where does it kill things off? Out of that, we get this, which talks about acute toxicity categories for pesticide products. This is in U.S. law, Section 40 of the Federal Code, 156.64 toxicity categories. The different toxicity categories, 1, 2, 3, 4 across the top, the method of application of the material. It gives you an oral LD50. It gives you an inhalation LD50. The oral is in milligrams per kilogram. The inhalation is milligrams per liter. The dermal LD50 is milligrams per kilogram. It looks at what happens if it goes in the eye, what happens if it's put on the skin. So up here on oral LD50, the bottom group says if the oral LD50 is less than or equal to 50, it is considered a toxic material category 1, which means you have to have special permit to be able to buy this. You aren't going to get this stuff. It's going to be labeled danger, poison, whereas a number 2 is going to be labeled just danger. Number 3 is going to be labeled warning. Number 4 is going to be labeled caution. Inhalation, you can see that these values are in milligrams per liter. You're breathing it in. These values appear to be much less than the upper ones. If you have inhalation damage, that can be very problematic at a very low level. Dermal LD50, you can see these values are about 4 times what you've got up on the oral part. The dermal part is much harder to get it in. When you get it on your skin, the first thing you do is wash and get rid of it. On the eye, they look at corrosive, whether or not it creates corneal opacity, whether or not the corneal opacity is reversible within a 7-day period. If it is, then they look at irritation and how long does the irritation persist for. On skin, you look at whether or not it's corrosive. That means you're going to get acid burns. You take something like sulfuric acid, that's corrosive. The part 2 looks at severe irritation at 72 hours. Then you're looking at moderate irritation at 72 hours. Then you're looking at mild or slight irritation at 72 hours. Either it causes damage very quickly or they look at what happens after 72 hours. That's a good indicator of how this is. When we do toxicology, we are trying to get all of this information so that we can make sure that the applicator is still in very good condition at the end.